So thanks, David, for the introduction. I think that's the first time now. I don't know how many years since, it's been 25 years or so since I finished in your lab, and first time I've had the pleasure of having you introduce me. And, so thank you. Also, thanks to the program committee for inviting me here to give this talk. This is a different audience than I normally speak to or usually get to listen to, so it's been really nice to hear a different perspective on things. And in that vein, I think I'm also going to try to give you a little bit different perspective on things. So actually, I had finished preparing my talk last night, and then over dinner I was talking to Fritz and thinking about an alternate introduction that I sometimes give to this talk, and I decided to cut back a little bit on the historical data and just give both introductions. So the first introduction starts with this slide and emphasizes what I think is probably one of the most important take-home messages, which is simply that you could have an entire wiring diagram of the brain, every single connection, and you wouldn't know how it worked. You could, take 20 of the best Russian physicists in 10 years, they still wouldn't tell you how the brain worked. They would quit and go off and sell derivatives on the stock market. And that's because we can't just take the circuit by itself, but we have to be able to link circuitry to function. And to do that, we need both correlations and causal experiments to test it. So even correlations by themselves, as you know, are just correlations, and you have to have ways to test whether hypotheses that come from those correlations actually hold up to experimental observations where you perturb the system. Okay? The next point that I want to make in this version of the introduction is that there are many different levels of resolution at which you can study circuits, and they're all useful, but for different reasons. And it's also important to think about what kinds of information you get from studying circuitry at different levels of resolution, and also what kind of tools you need if you're going to be able to understand what's going on at those different levels of resolution. So for example, historically, we've known for a very long time and have had good methods for looking at connections between brain areas, standard anterograde and retrograde tracers. You can inject in one area or another. You can find the same area in a different animal or the same species over and over again, and you can correlate that structure to the function by making recordings and seeing that those brain areas respond differently to different kinds of visual stimuli, for example. You can make manipulations like do lesions of a brain area and look at deficits in function. And those kinds of studies have been very useful at telling us what kinds of transformations go on between brain areas. You see a difference in the input to an area and the output. Okay? So that tells you a transformation that's going on, but what it doesn't tell you is how that happened. What are the neural mechanisms? What are the circuits that underlie that transformation, and how do they work? And to do that, you have to get to the microcircuit level. And in the next couple of slides, I'm going to tell you some about work that's been done in brain slices, which is an isolated preparation, to demonstrate levels of specificity that exist at the microcircuit level that point us to the kinds of methods that we're going to need to have, the levels of resolution we need to be able to perturb circuits or have correlative studies at the microcircuit level. But those studies that I'm going to tell you about that are done in brain slices don't link back to areas. So that's a big problem. It's very difficult to get microcircuit level information and link it back to connections that are much more distant. There have been people that have done heroic EM studies like Kevin Martin or Helen Barbas or Andreas Burkhalter that have done EM studies and get at little fragments of this, but it's very hard to do in a more systematic way. Okay? So that's something that we need better methods for as well. So if we go to a little bit finer level of resolution to individual neurons, a method that was used a lot in the 80s and early 90s in my lab and other labs was to intracellularly fill neurons with dye and reconstruct their dendritic and axonal arbors. So these are a bunch of neurons, just some selected ones from monkey primary visual cortex with dendrites in blue and axons in yellow. And you can see at this level of resolution, you can start to rule out connections. That is, you can see that, say, this cell sends axons that go right through layer 4C and arborize up here, so they're much less likely to make connections to whatever neurons have dendrites in that area. So this gives you what Misha Chishkovsky refers to as the potential connectivity. And you can even do things like Kevin Martin has done systematically to quantify all these axonal arbors that come into a place, and then look at all the cell types that, say, have dendrites there, and just guess that the connections are proportionate to the overlap of dendrites and axons, and you can make a circuit diagram that way. But what I'm going to show you in the next slide is that that prediction is wrong, and that when axons project somewhere, and there are multiple cell types that have dendrites there, they can selectively connect to some cells relative to other cells. And we first saw this clearly using the method that David alluded to, and I'm just going to show the result and not how the method works, where we recorded intracellularly from single neurons, stimulated neurons in different layers, and functionally measured the strength of connections from different layers. This is in rat visual cortex under these different cell types. And these are just representative examples, but for the different cell types, the same cell type would consistently have the same pattern of input. So here these pyramidal neurons and fast-making inhibitory neurons get a pattern of input that's close to what you might have expected just by looking at the density of axons that come from cells in these different layers. But these other cell types get a very different pattern. So here, this cell type gets a lot more input from layer 5 than superficial layers, and this cell type gets more input from superficial layers than deep layers. And we and other labs have looked at this kind of cell type specificity of connections, and it turns out to be much more the rule than the exception that connections are cell type specific. These are examples from inhibitory neurons, but it also turns out to be true for different types of excitatory neurons. So connections are cell type specific, and this is something that's going to matter. Unfortunately, as we'll come back to later, and as Alan Jones and also Ken Seguino mentioned a little earlier today, this is useful for us because there are tools, genetic methods for targeting cell types. So there is some hope for being able to go from animal to animal and be able to link connectivity to function at that level of resolution. A somewhat more difficult problem emerges, though, when you realize that neurons of the same cell type that are right next to each other are connected differently and can serve different functions. So if you have your hands on a particular cell of a particular type, and you look at that cell in one animal versus another animal, you're not going to be able to necessarily link the structure of the circuit to the function very well. So this is from published work about 6 years ago, showing that if I'm not sure if this audience knows, but if you just take two pyramidal neurons in the cortex that are right next to each other, they're usually not connected to each other. They're only connected to each other about 10 or 20% of the time. So the connections are fairly sparse. But it turns out that when they are connected to each other, they share common input. This input, say, from layer 4 is also sparse, 10% of the time they're connected. But when cells are connected to each other, they share common input that's reinforced by excitatory connections that are also selected within that subnetwork, and certain types of inhibitory neurons also make selective connections relative to those fine-scale subnetworks. So these sort of subnetworks embedded within a cortical column that are connected differently. So that's going to create a challenge if you want to link circuitry to function. You might have to do it in the same animal and not going across animals. Okay, so what we need are tools that can reveal connectivity and allow direct causal and correlative studies linking circuitry to function at the level of cell types and even single neurons. And as I said, we may be able to link function to cell types in separate animals. You can access the same cell types from one animal to another, but the unique contributions of a particular neuron might require linking structure to function in the same animal. So as we get deeper into this talk, what I'm going to do is show you how we can now do these things with the methods we've developed with rabies virus. So the second version of my introduction is in the context of connectomics. So I thought this might be interesting for this audience. Earlier this morning, with some of the talks, David mentioned the idea of a macro connectome. I suppose then if there are macro connectomes, there must be micro connectomes. And so it sort of tells us we got to think about what do we mean when we say this word, and to different people, the same word can mean different things. So I'm going to be careful about how I define that. And I'm going to talk about two different kinds of connectomes that we could potentially make with the Rabies-based tools that we've developed. But before we start doing things like making connectomes or doing large-scale projects, I think it's important to think about sort of doing a cost-benefit analysis and to ask about whether the data that we might collect is going to be usable and whether it's going to get us moving forward in a way that is commensurate with the cost in terms of understanding how the brain works. And that's not to say that we shouldn't always be pushing forward and making progress, because sometimes what you can do today isn't good enough, and only by moving forward and working harder at actually trying to solve the problem do you make your methods good enough so that the cost actually does match up to the benefits. But I'm going to, in talking about this, point out some examples from the past of different kinds of projects that had different costs and benefits. But so at any rate, why would we want a connectome? Of course, one thing that you might get from having a lot of information about connections in the brain is you might get information about certain organizing principles, or you might suggest functional relationships that would guide functional assays. And we know this from past examples of connectomes that already exist, like the complete connectome from C. elegans, or connections between primate visual cortical areas that we've already heard a lot about in this meeting. So for example, if we take this sort of partial low resolution connectome that's getting more and more complete with Henry's data and others, there are a number of principles that came out of this. One is that the idea of the hierarchy is an organizing principle. So of course, coming into this data set was not just information about there being connections, but also the laminar origins and terminations, as we've already heard about, that help to define this hierarchy. And also, these connections are always reciprocal, so that's a principle. And these observations guided a lot of functional assays. And those functional assays led to still more information about these hierarchies. So for example, if you lesion a higher area and look at the effects on a lower area, you see far fewer effects than if you lesion a lower area and look at the higher area, you see pretty dramatic effects. So that tells us, allows us to make predictions about what would have happened if you made a lesion in some other area that hasn't been tested before. So that's very useful to have these kind of organizing principles and have them linked to function with actual experiments, so we can predict what would go on with an experiment that hasn't been done before, so you don't have to test every single possibility that there is. Another connectome that's existed actually for more than 25 years is the complete connectome based on EM of C. elegans. And I actually think that until very recently, this connectome has been much less useful than the partial low resolution connectome. And that's because there wasn't a way to directly link the structure of the circuit to function. You can't stick an electrode in neurons in C. elegans, and there didn't exist until recently ways to then monitor the activity of specific cells, relate it to behavior, or to manipulate the activity of cells. But in the last few years, that's changed dramatically, and many labs like Corey Bargman's lab, Shrika Chalasani, who was with Corey and is now at the Salk Institute, are doing these really nice experiments that are allowing them to link circuitry to function. And one of the things about this complete wiring diagram in C. elegans that makes it so useful is that it's the same in every worm. So you can make observations of the circuit and do complete EM reconstruction in one worm, and you know that applies to all the other worms that you study. Okay. So they were a little premature in this, and probably what took them many, many years to do with the computers and ways to reconstruct we have now could have been done a lot more quickly. But nevertheless, now, 25 years later, it is quite useful to have this. And it's useful, again, I mentioned, because it's the same from worm to worm. But would you want to do a complete EM reconstruction of a mammalian brain, or say even a 1 millimeter cube of the mammalian brain? And I would argue that that would be not particularly useful because different mammalian brains are connected differently. So you might find organizing principles such as connection motifs, but this wouldn't allow you, it wouldn't guide functional assays, where if you had, the complete wiring diagram at the EM level of a piece of cortex, at this point in time, I think, wouldn't be as valuable as many other kinds of data that could be obtained. And in addition, we have to think about the cost and the benefit both, and think about what it would take to do this, and what are the difficulties in doing an EM reconstruction of something like a cube 1 millimeter of cortex. And right now, I think the technology is pretty far from being able to do that. We can guess that maybe in about 10 years somebody could get the complete wiring diagram, but the real difficulties lie in both identifying synapses. So if you have a nice pretty EM picture like this that's cut across synaptic specialization, and you can see pre- and post-synaptic specializations, there's a synapse there, but quite often the plane of section isn't such that you can identify the synapse, and so you can miss synapses. And the other problem that's very difficult to solve is to trace and follow the wires accurately, follow axons from section to section and reconstruct, and to be able to accurately tell the difference between an axon that bifurcates versus axons that cross. And as you go over larger volumes, this problem gets bigger and multiplies. Okay? So this then transitions to telling you more about the methods that we've been developing. And one of the things I want to say about rabies virus is that nature, through the evolution of this virus, has solved already for us exactly these two problems that are most difficult with EM, that is identifying synapses and tracing the wires. Okay? Because rabies virus naturally spreads only between neurons that have synaptic contacts, so it identifies synapses, and then the viral particles spread from the axon terminals up to the cell body and replicate in the cell, and then label that cell so we can trace the wires as well as identifying the connections. Okay? So then what can we do with this? I'm going to show you methods that we've developed that could allow you to make cell type connectomes by interfacing with mouse lines that have Crick-Jacobs or some other genes expressed selectively just in particular cell types. And I would argue that a cell type connectome is useful because cell types are conserved across individuals and species. So here's a chandelier cell, looks the same in a giraffe, as Javier de Felipe has shown, or a monkey or a human, and is likely to, at least at some level, have some common function. Insofar as we've looked at specificity of connections to different cell types, we see that this is conserved across brain areas and to different brains. We can target, as I mentioned, cell types with genetic techniques. So this allows you in the same mouse line, as Alan Jones mentioned before, to say, as I'll show you, trace connections, but then you can also say use optogenetic methods to manipulate the activity of a specific cell type, or express a genetically encoded indicator to correlate the activity of that cell type, its function, with the circuit. So I'll be saying more about that. Another type of connectome that one might think about making, and I'm not sure if it's quite as valuable if you just do it independent of function, is what I'll call a single cell connectome, and that would be the complete set of connections for a single cell. So with the rabies, and actually it's shown in this picture, you can label neurons that you know all must be converging onto a single neuron, and you'll see later how that works. And this could be useful, I think, for getting it. If you did this statistically over and over again for many cells of the same type, you get some statistical information, maybe get at some principles, but that by itself I don't think it would link you to function as well as you would like. And also the methods that we've developed so far based on rabies virus only label a subset of the inputs to a cell. So this would need still more development to get good enough for that. But one thing that the rabies virus, as I'll show you later, does is it labels connections to live cells. So in an intact animal, you can actually directly link structure to function for the same individual cell. Okay. So with that long double introduction then, I'll finally get to what we've done with rabies virus. And this is a figure from a paper we published 4 years ago, which was just a proof of principle paper that we could target rabies virus infection to specific cells and label the direct inputs to those cells. And this is a brain slice culture, so at this point in time we just had these methods working in slice culture and not yet in vivo. And what you're seeing here is there's one cell right here that's yellow. This is a blow up of it, so the red and green together make yellow. So we know from the red that this is what we call a starter cell, a cell that was the initial source of a rabies virus infection. So in ways that I'll tell you about, we were able to get this rabies virus infection to be targeted initially to just that cell. And then all of the green cells are cells that the rabies then spread to. And we know that they're rabies infected because we have green fluorescent protein in the rabies genome, so only rabies infected cells turn green. So rabies naturally, as I mentioned earlier, spreads just between connected neurons and exclusively in the retrograde direction. So the picture that you see here, where you see one starter cell and then green cells that are directly presynaptic to that cell, is very different than what rabies would do in nature if we just took normal wild-type rabies and put it on here. So rabies virus naturally infects axon terminals. Axon terminals have receptors for rabies glycoprotein. We'll see more about glycoprotein later. And so if we just put rabies virus onto a slice culture like this and it were normal wild-type rabies, we would have just non-specifically infected all kinds of cells all over the place. And then it would have spread not just across one synaptic step, labeling the direct inputs, but it would have continued to spread and we just have rabies infection all over this whole slice. Okay? So we needed to make modifications to the rabies virus to do two things: to get a specific infection and to control the spread of the virus so it would only go one synaptic step and then stop. Okay, so good. So essentially, what we wanted to do was to build a better rabies virus to infect specific cells or cell types. We wanted it to be able to maintain its ability to replicate, because if you have just a small number of particles that cross a synaptic contact, you wouldn't be able to detect those very easily. But if the virus replicates and expresses lots of genes from its genome, you can then detect that, like all these green cells here. And then again, we wanted to cross just one synaptic step and then stop. So the key to being able to do this was to use rabies virus that has its glycoprotein gene deleted. And in the next slide, you're going to see what I mean by that and how that works. But what that allows us to do is to do two things. First, to pseudotype the virus. I'll tell you what that means in the next slide, so that we could infect specific cell types. Doing this doesn't prevent the virus from replicating what's in a cell. It just prevents it from spreading, so we can control the spread of the virus if its glycoprotein gene is deleted. So here's what a normal rabies virus particle looks like. It's a negative strand RNA virus, has only RNA in its genome, replicates only in the cytoplasm and never goes to the nucleus. It has its RNA genome here inside this particle. And here's a schematic of the genome. Normally there are 5 genes, NPM, G, and L. We're just going to worry about G, the glycoprotein gene. What a glycoprotein is for this type of envelope virus is these little red spikes on the outside are the glycoprotein. So when the virus grows inside of a cell, like this one, it buds out from the cell and it takes with it the glycoprotein that was coded in its genome and got embedded in the cell membrane, and then this piece of the cell membrane envelops the viral particle, and it has these spikes on the outside, the glycoprotein. So it's this glycoprotein then that interacts with the axon terminal to mediate infection through the axon terminal. Okay. So if we delete the glycoprotein gene from the genome, we can replace it with green fluorescent protein, make viral particles that we grow in culture that look like this, and these will infect axon terminals, as you'll see in a slide in just a minute. And they'll infect, go back to the cell body, replicate, make lots of green fluorescent protein, but as you'll see, they're stuck inside the cell and can't get out. So we've been able, through that mechanism, to control the spread of the virus. Okay. But the other thing I told you we wanted to do is to make it so that this virus would initially only infect certain cells, and the way we do that is to pseudotype the virus. So if the glycoprotein gene is deleted, and we now in culture have cells that we infect, then there won't be any rabies glycoprotein expressed in these cells. Instead, we can express through other means a different envelope protein or glycoprotein called NV. And then what comes out are rabies viral particles that are on their envelope have a different glycoprotein. So this changes the infectious properties of the virus. And this particular envelope protein that we selected does not have any receptors in the mammalian brain. So a viral particle like this one we injected into a mammalian brain infects nothing. But we know what the receptor is for NV. It's a very nice, tight association with a receptor called TVA. So we can simply express TVA in cells that we would like to have infected with this virus, and then that's what gives us our selective infection. Okay. So I'm going to tell you about experiments we've done to use this kind of rabies virus. We call it delta G. It's a glycoprotein-deleted rabies virus to look at brain circuitry. And first, to illustrate that deletion of the glycoprotein prevents spread of the virus, I'll also show you that it works as a really nice retrograde tracer. And one of the nice things about it is that all in one step, you can inject it into some part of the brain. It infects the axon terminals, goes back to the cell, and it completely fills the cells with GFP. Here you can see cells that are projecting to superior colliculus, others that are projecting, I think this is to the thalamus, but not just to a particular thalamic nucleus. And this is all in mouse, and the cell types that we get labeled based on the different locations we inject are what we expect from previous studies, but they're nicely filled. And you can also reconstruct the axons of these cells if you look at higher power. But you can see that here, these are only the cells that project to the colliculus, only that cell type, these tall tufted cells, and other cells that we know connect to them are not labeled, so the virus doesn't spread. Okay. So next what we want to do is to now use our NV pseudotype virus to target neurons and then label their direct input. So how can we do that? And I'll show you that first in the context of doing experiments in vivo to label inputs to a single neuron. And this is work that Jim Marshall, who just defended his thesis last week, did in the lab. And this illustrates schematically what we want. So the way you do this is to start with, you first have to access a single neuron, and the way to get that started is to electroporate DNA into a single cell in vivo. And we use methods that were developed in Michael Hauser's lab called shadow patching, which allow us under two-photon visualization to see our electrode go toward the cell and electroporate DNA into that one cell. And the plasmids that are put into the pipette code for 3 different genes. First, just a yellow fluorescent protein to mark the cell so we'll be able to find it later. TVA, remember that's the receptor for NV, so the rabies virus will be able to infect that cell. And the rabies glycoprotein, as you see, that's going to be necessary to allow the virus to spread out from the cell. So you wait a few days, these genes are expressed, so now they're proteins. The cell's green, has glycoprotein that's actually in the membrane, but it's illustrated in the cytoplasm here, the TVA receptor. So now if you inject an NV pseudotype rabies virus, in this case with M. cherry in the genome, so infected cells will turn red, that will selectively infect that cell and replicate inside the cell. It takes advantage of the rabies glycoprotein that's in the cell, so that allows it to spread from this cell to the directly presynaptic cells, which will then turn red. And now these cells are infected, have lots of copies of the virus and nice bright red, but there's no glycoprotein, rabies glycoprotein in these cells, so the virus can't get out and spread any further. Okay. And this is the timeline of these experiments. You electroporate, wait 2 or 3 days, infect with the virus, and wait roughly a week for the virus to spread to presynaptic neurons. And these are just some pictures of one of the cases where this is an example where the electroporation included M. cherry and the rabies virus is going to express GFP. So this is a layer 2-3 pyramidal neuron and mouse visual cortex, a blow up of that cell. This is postmortem histology after perfusing the animal, making sections. And if we add in the green channel, you can see rabies-infected neurons in vivo. The blue is a counter-stained DAPI. So you can see many neurons get labeled that are directly presynaptic to that one cell. And you can go through adjacent sections and find more and more cells, and even find cells very far away that have been infected and are presumably connecting to that cell. So at this point, this method works as well as the electroporation does. Every time we have successful electroporation, you have successful infection and successful spread of the virus. One of the limitations is that the numbers of neurons that we're infecting is almost certainly a small proportion of all of them. We don't know how many inputs there should be. We label up to 100. We think there should be on the order of 500 to 1,000 inputs to a single neuron like this. So we're working very hard on improving this and getting more inputs. Already this is, I think, very valuable for things like directly linking structure to function. So as you'll see later in the talk, we now have ways to image the activity of neurons that have been infected with the rabies virus and look at their functional properties. So you might ask a question like, well, we know the rules of fine-scale connections are different for layer 3 cells that connect to this cell than for layer 5 cells. And we think from our work and others that layer 5 subnetworks are orthogonal to layer 3 subnetworks. So we would guess that these neighboring neurons in the visual cortex in layer 3 have similar orientation selectivity to their starter cell, but layer 5 cells might have some other functional property, and knowing what that is would help us to make inferences about the relative importance of connections from layer 5 versus layer 3. Next, I'm going to tell you about how we can label the direct input to a population of cells of a single genetically targeted type, and this is now taking advantage of mouse lines that have Crick-Jacobs in a specific cell type. This is work that Nick Wall did. He's a graduate student in the lab, and the basic idea, which is illustrated here, is that you're going to start with a mouse line that expresses Crick-Jacobs in a specific cell type, so that's this cell here. You're going to use then what we call a helper virus, which you inject into the region, and it's going to indiscriminately infect any cell here, but only the cells with Crick-Jacobs will express the genes that are carried by that virus. They'll be the same genes that we electroporated in the single cell example, TVA and rabies glycoprotein, and a marker gene, and then we can selectively infect these cells of this type with the N-based pseudotype rabies virus, get the complementation through the glycoprotein that's expressed from the helper virus, and then spread to and label the presynaptic inputs, but no multisynaptic spread. So this is just an illustration of the gene construct that's in the helper virus. In the experiments I'm going to show you, we use an adeno-associated virus, and it has what's known as a flex construct, so it's a reverse open reading frame with a promoter here. So in the absence of Crick-Jacobs, you don't get any gene expression, but following Crick-recombination, these lock sites are oriented so that this gets flipped into the correct open reading frame, and this excess lock site here gets removed so that now it can't flip back, and so you get these genes expressed only in the Crick-expressing cells. And in these experiments, because adeno-associated virus takes several weeks to get good high levels of gene expression, you need to first inject the helper virus. We wait 3 weeks, then inject the rabies virus, wait a week for the spread of the rabies from the starter cells to the presynaptic neurons, and then perfuse the animal, and then look at the gene expression. So in this first example, we used a line of mice called L7-Cre, which expresses Crick-recombinase mostly, although not entirely, in cerebellar Purkinje cells. And here, this is a control experiment where we first use a helper virus that, following Crick-recombination, expresses green fluorescent protein and TVA, but no rabies glycoprotein. And if we use that helper virus in the absence of rabies, you can see the GFP is expressed just in the cerebellar Purkinje cells as expected. And now, if we add an M-cherry expressing rabies that's N-based pseudotype, it selectively infects those GFP-positive Purkinje cells, but it doesn't spread transsynaptically because there's no glycoprotein in this helper virus. So this illustrates the selectivity. In a wild-type mouse, there's no TVA expression, and so no infection with the rabies. And in the absence of glycoprotein, there's no spread. So in the next slide, if we do the same experiment, but now use a different helper virus that also expresses rabies glycoprotein, and in this early version, we had a problem with the expression of the GFP that we've now fixed, but so there are newer versions of this. If anybody's interested, we can get those to you. But at any rate, now that you have TVA and rabies glycoprotein, in addition to all the cerebellar Purkinje cells, you have spread of the virus to direct presynaptic inputs. All these little spots here are granule cells. All these are the axons, the parallel fibers that connect to the Purkinje cells from the granule cells. And then there's the climbing fiber input that comes from the inferior olive up here. And so you can see that we get transsynaptic spread of the virus in the presence of the complementation and input from the expected locations. Here's another example using a parvalbumin Cree mouse from Sylvia Arbor. This is in the barrel cortex, and here again we use this helper virus that's got TVA and rabies glycoprotein, and if we looked close here, we would see, and with antibody staining, we'd see, in addition to the parvalbumin positive inhibitory neurons that are starter cells, there are many pyramidal cells that make local connections to those inhibitory cells. Here you see a failure to some extent of our control, that there is some leaky TVA expression in the wild type animal. This is something we've now improved and cleaned up in various ways, but mostly that's just, we use a different method in a mouse line to get the TVA expressed more selectively rather than from the helper virus that leaks a bit. So this affects the local interpretation, but there's no transsynaptic spread. The leak is not enough in terms of the glycoprotein to get transsynaptic spread. So when we look at more distant structures like here, we can be confident that these are direct inputs to the parvalbumin cells. And they're in the expected locations, such as the S2 barrel cortex, connecting to those cells. And then here in the thalamus, we see two clusters of cells that correspond to two thalamic nuclei that are known to project to the barrel cortex. They are PO and VPM. We knew from previous studies that functional studies showing that VPM connects to them, but it wasn't necessarily clear whether PO would. And then more recently, we've taken advantage of mouse lines that Josh Wong has made that have Cre recombinase in different types of inhibitory cells. So somatostatin expressing inhibitory cells that are these Martinotti cells, VIP, and then also the parvalbumin. And those are actually turn out to be three separate populations of inhibitory cells that are non-overlapping. And if you look then at inputs to the barrel cortex from S2 or from thalamus, we see that these different populations of cells have differences in their input. And it turns out that we don't detect input from S2 or thalamus to the somatostatin Cre line. These are somatostatin cells, and that's consistent with previous functional observations from Barry Connors, where if they record from what they call low threshold spiking cells and electrically stimulate the thalamus, they don't see input to these cells, but they do see it to the fast spiking cells here. And then here, interestingly, for the VIP Cre cells, we see input from the barrel cortex, but not the thalamus, suggesting that the parvalbumin cells may be the exclusive recipients of thalamic input, or at least in terms of functional strength, they're probably much more dominant. Okay, so finally, I told you that ultimately what we want to do is to link connectivity to function. We don't want to just look at these connections structurally. And so what we've been doing over the last couple of years is to take advantage of the fact that the rabies virus, once it infects cells, it will eventually kill them after about 2 weeks, but there's an extended period of time where they're viable, and you can do functional studies. And so what we can do is to insert different genes into the rabies genome. This is just an example of blue fluorescent protein, or multiple genes into the genome. And we can start to interface this tracing method with the other molecular and genetic tools that have been developed. So this is just a partial list of some of the genes. This paper was just published a couple of weeks ago that we've put into the rabies genome, and we have tools that are are deposited at AG in all these plasmids, so anybody wants to insert any other gene they'd like to think of. But here, as I'll show you with GCAMP3 expressed from the rabies genome, we can monitor the activity of cells that have defined connections or control their activity with channel rhodopsin, a latostatin receptor. We've now put archirhodopsin in. Or we can also control gene expression. There are a lot of people doing developmental studies that might like to turn on or off a gene in connectually defined cells. And this is just an example of the rabies that expresses an M-cherry channel rhodopsin 2 fusion protein. This is an injection in the barrel cortex and region selected with sparse expression so that we could then target intracellular recordings to those cells and just demonstrate that, as you expect, if they express channel rhodopsin, you can flash blue light and reliably generate action potentials in the cells. This is a biocytin fill. This is showing just that we recorded from a cell that was red, so that works as expected. Experiments that we've been working in the lab over the last year or so, we've been doing a lot of work functionally characterizing the 9 different extrastriate visual areas that Andreas Burkhalter has shown surround V1, so we can map those areas with intrinsic signal imaging and functionally characterize those cells. But here what we did is we wondered what are the sources of the functional differences between extrastriate areas? So we'd like to measure the visual responses of neurons in V1 that we know project to a particular cortical area. So after mapping with intrinsic signal imaging, what we can do is inject a rabies virus that expresses GCAMP3 and DS-RED that helps us to find the cells in neurons that project to that location. So after infecting, we come back, and now with 2-photon imaging, we can make this structural image in the live animal, and this is a series of Z scans coming down here. And it actually turns out that we can image remarkably deep into the cortex with this method. We're not quite sure why this is possible. It's not possible with Oregon grain Bapta in our hands, but with the rabies virus, which is sparse and high levels of expression, we can image down more than 1 millimeter. And then we're able to functionally characterize cells not only in the superficial layers, but deep layers. So here's an example from, I think, around 300 microns deep, where we can see neurons that responded to drifting gratings in front of the mouse that are, in these 2 cases, direction-selective neurons. You can also select out dendrites and look at their calcium changes in response to visual stimuli. So this allows us then to directly link the connectivity of neurons to their functional properties, and this is something we're going to be exploiting a lot in the next year or 2. So that's all I have to say. I just want to end by showing this slide, which acknowledges the people who have done the work. I've tried to point them out as we went along, but I do want to mention Ian Wickersham, who at the very start worked on developing the whole approach to the transsynaptic tracing method. I mentioned other people in the lab as I went, but also John Young, who was a virologist at the Salk Institute, who was instrumental in pointing us in the direction of N-based pseudotyping for targeting cells. Thanks.